Hello and welcome everyone. In this podcast we're going to talk about the cell. We're going to talk about special characteristics of the cell. We're going to talk about what makes certain cells different from other cells. We're going to talk about the function of cells, the function of things that occur in the cell. And to get started, I want to talk about this very important concept in biology. It's a pretty strong foundation of biology, and it's called the cell theory. Well, your book does what I would say a fairly poor job at demonstrating what this is. So if you noticed in your note-taking guide, I pretty much just told you what this is. But let's say it again. The cell is the basic structural and functional unit of life. You know, when we talked about proteins, and we said proteins are made up of amino acids. And when we talked about nucleic acids, we said they're made up of nucleotides. When we think about life, life is made up of cells. You have to have a cell. This is the basic unit of life. The second tenet of the cell theory is that all organisms are composed of one or more cells. Organisms like bacteria, yeast, these are one-celled organisms. Other organisms, like a tree, an elephant, a human, are composed of more than one cell. But an organism has to have at least one cell. The third tenet, all cells are produced by the division of pre-existing cells. Make sure that you commit this cell theory to memory, these three basic tenets of it. Next, I want to list features shared by all cells. The first feature is that plasma membranes enclose the cell. In the cell is the cytosol. This is a semi-fluid liquid that fills the inside of the cell. That everything else inside of the cell kind of floats in this cytosol. The third thing common to all cells, chromosomes are present and they carry genes. The last thing here is they all contain ribosomes, which are important in building proteins. Next I want to talk about is a comparison of prokaryotes to eukaryotes. I'm going to write the word prokaryote here, but in a moment I'm going to replace that, because the word prokaryote is not the best word to use. Your book uses it. You may or may not have learned that term in high school, but it's a word we would like to get out of your vocabulary. There was a time when we felt that in this group of prokaryotes there were bacteria and another group called archaea. And we thought these were very similar to each other. They're small. When you look at them under a microscope, they appear very similar. They lack organelles. They're single-celled. Unlike the eukaryotes, which were larger, often multicellular. They contained organelles. So it seemed reasonable at first approximation that this term prokaryote included both bacteria and archaea, which came before eukaryotes. However, we now know that's not true. We now know archaea are actually more similar to eukaryotes than bacteria. So we'd like to stop using this term prokaryote. It's an outdated term, and it implies that these two, bacteria and archaea, are similar, or at least more similar to each other than to eukaryotes. I'm going to draw this in a different way, and this is a way we represent, or can represent, how organisms diverge from each other. We know that at this point here in evolution, there was a common ancestor to all of life. We know that bacteria were the first to form. They're the most primitive. The next thing would have been these archaea. And then finally, eukaryotes. And we can break these down a lot more, and we will later on towards the end of the semester. But for now, what I really want you to focus on is the fact that bacteria and archaea are separate groups, and they should not be lumped under prokaryotes. When we do a genome investigation of archaea and eukaryotes, we recognize and we realize that archaea and eukaryotes are much more similar to each other than either eukaryotes or archaea are to bacteria. So bacteria truly belong by themselves here as the most primitive, the first cell that formed. And archaea deserves to be its own separate group as well as eukaryotes. And when I say groups, what I really mean to say are domains. So we have the domain of bacteria, the domain of archaea, and the domain of eukaryotes. And they really are separate groups. I know I'm kind of belaboring this point, but as you move forward, it's best to refer to them in this way and to not refer to the term prokaryote. Even though, unfortunately, it's still on my note-taking guide as prokaryote, and your textbook still uses the term prokaryote. Sometimes it takes a while for textbooks to catch up. What I'm going to do now is talk about some features that are common to bacteria. What characterizes a bacterium? Again, your book will use the term prokaryote, but we're going to call them bacteria. We're not really going to talk about archaea too much right now. We'll come back to them later on. They're very interesting. But for right now, we want to just talk about bacteria. And let me draw kind of a bacterium here. They can be one of several shapes. I'm going to draw one that's shaped like this with flagella coming off of it. Now, some common features of bacteria is that they're pretty small. They're 1 to 10 micrometers. Not very large at all. Certainly can't see it without the aid of a microscope. We think of these as being the first cells to evolve. First cells to evolve. There are no organelles. I also like to add something here and say there are no true organelles. In some species of bacteria, we see these primitive types of organelle-like structures, but no true organelles that we think of when we talk about eukaryotes. So based on that, there's also no nucleus. It still has DNA, and it can't be in the nucleus because it doesn't exist, and there's no membrane structures like that in a bacterial cell. But the DNA is in what we call a nucleoid. Now it's true, if we were to talk about archaea, many of these same things we would see. Archaea also have our 1 to 10 micrometers in size. They weren't the first to evolve. They also lack true organelles. They don't have a nucleus, but they do have these DNA nucleoids. Alright, that's all you need to know about bacteria for now. We will come back to this towards the end of the semester. So let me erase this, and let's talk about eukaryotes. Eukaryotes include protista, fungi, plants, and animals. Their size is anywhere from 10 to 100 micrometers. They're quite a bit bigger than the bacterium. They share a common ancestor with archaea and bacteria. They contain organelles, and these organelles each have a discrete function. Just like bacterium, they have DNA, chromosomes, but this time in eukaryotes, these are stored in a nucleus. Now for the rest of this podcast, we're going to focus on eukaryotes because we want to talk about specific organelles. There may be some things that are common also to bacterium, and when those come along, I will make sure I make a note of that. So let's talk about the plasma membrane. If we were to draw a cell, you could draw it like this. The plasma membrane is on the outside. I'm going to draw it sort of like this. And that's because the plasma membrane is a double layer of phospholipids. And other molecules. Now I'm not going to draw this all the way across, I'm just going to draw it on part of this. But remember, the phospholipids, we have this polar phosphate group, or hydrophilic phosphate group. And then we have these hydrophobic fatty acid tails. And we're going to have this on both sides of the membrane. This is why we call it a double layer. We will sometimes call this a bilayer. Alright, I'm just going to draw a couple more of these. We also know that in addition to these phospholipids, as I wrote here, other molecules, that various proteins can be stuck in these membranes. So I'm just going to write protein. These turn out to be very important. We also know within the membranes, and we'll come back to this later, we could have various kinds of hormones. I'm just going to put a C here for cholesterol. Sometimes we can have that in our membranes as well. The main purpose of the plasma membrane, we can think of it as having a couple functions. It does serve a role in protecting the cell, and it serves as a selective barrier. Meaning this cell will not allow everything into the cytoplasm. Only limited things can pass through this membrane, and that's because of this selective barrier. This is very important. Now, I said I would point out things that are common in both eukaryotes and bacterium, and this would be one of them. We see this plasma membrane in both bacterium and eukaryotes, as well as archaea. Next I want to talk about is the cytoplasm and the cytosol. I really want to take a moment to distinguish the difference between a cytoplasm and a cytosol. We often use these terms interchangeably. Incorrectly, we shouldn't do that, but we often do use them interchangeably, and you will catch me mistakenly using them interchangeably too. But this is how they are different. So the cytosol is the intracellular fluid surrounds, or you might say bathes, the organelles. Most of this is going to be water plus various ions and molecules that have been dissolved in the water. So for instance, if I were to draw a cell here, if we just looked at the inside of the cell, we would call that the cytosol. So what makes it the cytoplasm, as we have here, was when you take the cytosol and you add to the cytosol organelles. So for instance, if this was our cytosol here, and now let's start putting some organelles in. Let's put a nucleus in. We'll put a couple peroxisomes in here. Let's put some mitochondria in here. Some ER, like so. And let's draw some Golgi stacks in here next. And there are other organelles. But the point of the matter is that it's these organelles plus the cytosol that make up the cytoplasm. If what you're looking at doesn't have the organelles in it, you just call it cytosol. I'm going to erase this because I want to spend just a few minutes talking about some general features of organelles. On the last slide, I showed you a generic picture showing some of the organelles, but what are their general purposes? Why do we have organelles? Well, first, let's recognize that they are membrane enclosed compartments. Next, they contain, within this membrane enclosed compartment, they contain specific enzymes. And these specific enzymes help give that unique function to that particular organelle. Each organelle has a different function. And that's this next point here, is that each organelle performs a specific function. The numbers and types of organelles varies per species and tissue type. For instance, if you had a cell that required lots of energy, you would find a lot of mitochondria in it. If it was a cell that didn't require as much energy, there would be fewer mitochondria in it. So this is a dynamic number. Alright, now let's spend some time talking about specific organelles. Now, as we go through each of the parts of the cell, what I'm going to try to do is maintain this large cell over here. We've already talked about the plasma membrane, so let's put that in here. And we've also talked about the cytosol. The first organelle we want to talk about is the nucleus. And let's go ahead and put the nucleus right here. And I'm going to put an N in here for nucleus. What is the purpose of the nucleus? Well, its function is that it holds, you could say, chromosomes. Remember, chromosomes, this is where our DNA is, including our genes, of course. Within the nucleus, there's this special compartment which is called the nucleolus. And I'm going to put that in red here, and I'm just going to put NU. So it contains the nucleolus. And the purpose of the nucleolus is that this is where ribosomes are made. Now, ribosomes would be found in bacteria, archaea, and eukaryotes. I'm going to go ahead and just put a couple little dots here just to show that there are ribosomes. So down here I'm just going to put a dot and say that's a ribosome. Another thing I'm going to point out here is that the nucleus, like the mitochondria and the chloroplast, is double membraned. I'm going to put that here so it's under the blue area here. Double membrane. We'll talk more about ribosomes, but let's just say a couple more things about them. Ribosomes, and I'm just going to list that here kind of separately. Their main function is to make or synthesize, we could say, proteins. They are made up of proteins plus ribosomal RNA. And this is really all I want you to know about ribosomes. That they make proteins, they're made up of proteins and RNA, and finally they're found within the cytoplasm of a cell. Alright, let me erase this information about the nucleus and move on to our next organelle. So I want to talk about the endoplasmic reticulum next. Often we will just call this the ER. We have two kinds of ER. It could be the smooth ER or the rough ER. Let's talk about the smooth ER first. It has several functions. It synthesizes lipids that the cell will use. It metabolizes various carbohydrates. A very important role for it is that it detoxifies drugs and poisons. And lastly, it's a place where calcium ions are stored. We call it a smooth ER because there are no ribosomes attached to it. I'm going to draw it right here, kind of around the nucleus, like so. Now let's spend a little time talking about the rough ER. It has a few main functions. One is that it secretes proteins. What I mean by that is sometimes the cell will make a protein and it will kick it out of the cell, or we say it secretes the protein. The rough ER makes glycoproteins. A glycoprotein is like any other protein that we talked about when we talk about proteins as a macromolecule, except it has a sugar attached to it. And that's symbolized by saying glyco in front of it. The rough ER finally makes membranes. And down here I'm going to say it's not really a function, it's a characteristic, but it contains ribosomes. And so I'm going to just draw them over here on this side of the nucleus. And I'm going to litter it here with ribosomes. Okay, before we move on, as you look at your own drawing, you may want to label these as rough ER and these as smooth ER. I just don't have the space to do that on this small whiteboard here. All right, the next thing I want to talk about is the Golgi Apparatus. The Golgi Apparatus is very important in modifying proteins. So it modifies proteins. It can add various kinds of sugars to these proteins, and typically we're going to find these Golgi apparatuses localized close to the rough ER. Often the proteins that are made in the rough ER get shuttled into the Golgi, so they're often localized close to the ER, the rough ER. The Golgi apparatus also synthesizes polysaccharides. I should also mention that the part of the Golgi closest to the ER we call cis, and the part that's furthest away we call trans. Now you're going to get a lot more of this when you take a cell biology course, but what you should know, or just keep in the back of your mind, is that vesicles move from the ER to the cis, and then from the cis to the different other layers of the Golgi until ultimately they are released as a vesicle containing a very special protein that was made through processing it through the ER and the Golgi. Okay, that's all I want you to know about the Golgi. Let's move on to the lysosome. Now, the lysosome has a pretty interesting function. It sort of serves as the garbage collector of the cell in some ways, because it breaks down ingested substances. So things that are on the outside of the cell that get brought up into the cell are taken into the lysosome where they are degraded. In addition to breaking down ingested substances, it could also break down old or damaged organelles. They contain very strong digestive enzymes. Within the lysosome, it is a very acidic environment. So, let's draw our lysosome. And we will typically draw these as just circles like that. And they might be in different parts of the cell. These are our lysosomes. On your drawing, I would recommend going ahead and labeling those as lysosomes. The next thing I want to talk about are vacuoles. I think we could maybe best summarize vacuoles as being a vesicle in the cell that transports solutes. And there are different kinds of vacuoles. For instance, there are food vacuoles. They will bring in larger substances through phagocytosis. There are also vacuoles that we call contractile. These are the ones I really want you to know. The contractile vacuoles are important in transporting water in and out of the cell. This helps maintain a proper balance of ions within the cell. And one other vacuole that I want to mention, I don't have room, let's see, I'm going to put it above food vacuole, and these are called central vacuoles. You should know these as being vacuoles that we find in plants. These will also bring in water, but they're very important in allowing these plants to grow. And vacuoles tend to be quite large in a cell, particularly like the central vacuole. So what I'm going to do is I'm just going to kind of draw it like this as a dotted line here, kind of sitting over the top of all of the cell here. Okay, let's erase this. And I would mention this term here called the endomembrane system. We could think of the endomembrane system as a series of organelles and cellular parts that work together to regulate protein traffic in the cell. It does a few other things, but this is the most important thing for right now. So for instance, everything, or at least most of the things that we've drawn to this point would fall into this category. So for instance, the nucleus is where protein traffic starts, right here. It will make a messenger RNA that we'll talk about later, and then it'll insert into the rough ER. It will then pass this protein from the ER to the Golgi. From the Golgi, it could go to either a vacuole, the plasma membrane to export it outside of the cell, or the lysosome to have that particular protein either degraded in the lysosome, or maybe it's the protein that needs to function in the lysosome, like one of those digestive enzymes. All of these organelles are important in the endomembrane system for trafficking proteins. Alright, the next thing I want to talk about are mitochondria. Now, often in textbooks, they draw mitochondria as these kind of oval, bean-shaped structures. And so I can't draw them that way because I know they're not shaped that way. I'm going to draw them a little bit like how they're really shaped. And they're usually located near the outside of the cell, like so. And they're usually long and tubular. If you want to know why the textbooks always draw this wrong, I can tell you later. And sometimes they're branched like this, allowing various mitochondrial tubules to extend from them. But what is their function? Well, they are the site of cellular respiration. Because of this, this is where a lot of the cell's ATP, the cell's energy, is made, right in those mitochondria. They are found in nearly, the mitochondria is also responsible for making parts of some vitamins and amino acids. It's also an incredibly important organelle in regulating something that is called apoptosis. Apoptosis is a program that the cell sets into place to trigger cell death. At first you might not think cell death is a good thing, but it's important that our cells recycle when it's time for them to recycle. They are found in animals, protists, fungi, and plants. Now the reason I list this here, we could talk about all the organelles and make the same listing here, but one of these things that kind of gets set in some people's mind is that mitochondria are not in plants. Well, that's not true. Plants are full of mitochondria. They require mitochondria because the plants also need to generate energy through cellular respiration, and they use the mitochondria for that, just like we use our mitochondria. After I erased that, I forgot one more important thing I want to say about mitochondria, and that is that they contain DNA. Just like the nucleus contains DNA, so do the mitochondria. Alright, next thing I want to talk about are chloroplasts. I want to mention them as I talk about mitochondria because, like mitochondria, chloroplasts also have their own DNA. What you're probably familiar with is that chloroplast is the organelle where photosynthesis occurs. And unlike mitochondria, which are found in all eukaryotes, chloroplasts are found in plants and photosynthetic protists. I'm going to draw the chloroplasts in here as well, and I'm going to draw them up along here. So these are our chloroplasts. The last organelle I want to talk about are the peroxisomes. Some of the things they do is that they perform oxidation reactions. That is, they break down fatty acids. They also produce hydrogen peroxide. They are usually small structures, circular structures. I'm going to draw them like so, these small circular structures. And they could be in different parts of the cell, so I'm going to put them in different parts of the cell. And the next thing I want to talk about is the cytoskeleton. It's not an organelle because it's not surrounded by a membrane, but the cytoskeleton is a very important cellular structure. They provide structure and support for the cell, provide motility, well, cell motility, that is in the cell, meaning moving things in the cell from one part of the cell to another part of the cell, and the whole cell. So in general, that's what the cytoskeleton does. But there are a couple specific cytoskeletons that I want to talk to you about. The first one are what we call microtubules. And the other one I want to talk to you about are called actin, or sometimes we call them filaments. Microtubules are thicker. They are made of this protein called tubulin. One of the things they do is they move chromosomes. So within here, I just put a little asterisk here, there's a lot of microtubules within the nucleus to help move chromosomes during cell division. We also see microtubules in cilia and flagella. So you might have cilia on the outside of the cell that may help direct movement of food or fluid around the cell. And the flagella may be at the end, one end of the cell to help allow movement of the whole cell. Alright, actin or filaments, actually better to call them microfilaments. So actin or microfilaments, these are thinner. Like microtubules are made up of tubulin, microfilaments are made up of this protein called actin, and that's why sometimes we call them actin filaments. They're very important in maintaining this cell shape, so you may see a network of actin cytoskeleton networked across the cell to help maintain that proper shape. They also play a role in cell division, and I'll write it up here, muscle contraction. There is one more kind of cytoskeleton that I want to tell you, and I don't have room to write it here, so I'm going to have to erase this. And this third cytoskeletal component is called an intermediate filament. They have several kinds of functions. We'll just list two here. Just like the actin microfilaments, they play a role in cell shape, in maintaining that cell shape, and they also play an important role in anchoring organelles, holding them in place at certain times. Now, if you recall, I mentioned that these cytoskeletal elements, and what I want to do here is draw a microtubule just as a straight line, and I'm going to have it picking up this lysosome right here. This blue lysosome. I believe we call it the lysosomes, the blue color here. But what I want to talk about next is something called motor proteins. When you take cell biology course or a physiology course, you'll learn a lot more about these motor proteins. And there's many kinds of these motor proteins that you'll need to learn. But for now, I want you to know that this cytoskeleton here, as I mentioned before, it can move organelles, and the organelles just don't randomly move, they require energy. And that energy is provided by these motor proteins. So these motor proteins are important for transporting things on the cytoskeleton, and they use ATP to generate this work. So know that motor proteins are important in helping the cytoskeleton move things across the cell. So what have I drawn here? Is it a plant cell or is it an animal cell? Well, there are features here that might make you think it's a plant cell, and there are features here that might make you think it's not a plant cell. These are the things common to plant cells. They have that central vacuole, which I indicated with this dotted line. They also have chloroplasts, which are up here. So that's really enough to call it a plant cell. But what I didn't draw, and I'm going to have to erase these cilia as well, and this flagellum, so we can make this a plant cell. Plant cells also, on the outside of the plasma membrane, they're enclosed by a cell wall, a very thick material that helps protect the plant. Now, animal cells don't have this cell wall, but what they do have is an extracellular matrix. And what you should know about the extracellular matrix is that it provides structural and biochemical support for surrounding cells. So as two cells are adjacent to each other, they will secrete various chemicals and various proteins that are needed for adjacent cells to function and coordinate properly. Now, the only thing I haven't talked about that's on your note-taking guide is this thing called the Endosymbiotic theory. Now what I want you to do is look in your textbook and I want you to read about the endosymbiotic theory and see if you can figure out exactly what it is and why it's important. And I'll give you a hint, as you'll quickly learn as you read it, but it has a lot to do with how eukaryotic cells have mitochondria and chloroplasts. Alright, that is all I have for this podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.